My talk is about the medical uses of mRNA-based directed gene delivery. The emphasis is on exosomes or extracellular vesicles as delivery vehicles. As you can see, I'm AC Matin, etc. This work has involved a number of collaborators. Some of them are represented on the slide. They include my colleagues at Stanford, some private capital companies, as well as people at NIH. This research was supported by NIH US2, US3 grants, Extracellular RNA Consortium, and CATS. Specific targeting of diseases is possible because receptors and ligands are overexpressed in many diseases. One example is the HER2 receptor. In HER2 positive breast cancer, it is highly overexpressed in this cancer. You can see that it is in a different color because it is this one that my presentation will concentrate on. But as you can see, if you go down the list, there are so many other receptors, ligands, and such that are expressed in other cancers as well as in other diseases listed below the dotted line. The focus of this talk is on prodrug-mediated cancer. I call it cured because I think we have achieved that without side effects. The topics are the background of prodrug treatment of cancer, and the merits of mRNA versus DNA as gene delivery agents, exosomes for gene delivery, their advantages or disadvantages, their loading with mRNA, making them directed, and then preclinical studies on their efficacy. And very importantly, emphasis on the importance of systemic administration. These things will become clear as I continue. If I have time, I'll briefly discuss similar use of lipid nanoparticles, which have had a long history of use, particularly these days with the coronavirus around. Their vaccines are very popular and useful. Okay, so what are prodrugs? I'll concentrate here on nitro prodrugs. They are inactive in their native state, but become highly toxic upon reduction, catalyzed by bacterial or a viral enzyme. The approach is termed gene-delivered enzyme prodrug therapy, or GDEPT for short. They kill cells by generating DNA adducts. They target both growing and non-growing cells. And I have changed the color of non-growing because I indicate why that is important later on. And thus they are better than drugs that target only growing cells. Examples are CB1954, the clinical name of which is Tretasecar, and then CNOB and its activating enzyme CHRR, which we discovered in my laboratory. Here's the background of GDEPT clinical trials, two of them. The most extensive study was done with genciclovir, using a viral enzyme, herpes simplex virus, thymidine kinase, which converts it into genciclovir triphosphate, which can muck up the DNA and kill the cell. This was tested in a phase 3 clinical trial with 248 glioblastoma multiforme patients. The gene was introduced using a retroviral vector, which was injected via craniotomy. And then the GC was administered intravenously, but there were no benefits. So the thing was a complete failure. The other one is the CB1954 Tretasecar, using the NTR, nitroreductase enzyme of Escherichia coli, and adenovirus vector was used to deliver the NTR gene directly into localized prostate cancer. Again, there were some benefits, but not really impressive enough to justify promotion to clinical trials. A lot of analysis was done post these trials, and it became clear that an important reason is that the wild-type enzymes are not potent enough. There is insufficient gene delivery and expression. The viral vectors can be problematic, immunogenicity, barrier to repeat dosage, pathogenicity. And there was no selective targeting of cancer, so administration had to be done at the cancer site. As we all know, not all cancers are accessible for direct injection, least of all cancer metastases. So we have attempted to address these issues, and this has been greatly helped by our discovery of the prodrug CNOB, which I just mentioned, whose activated product, MCHB, is fluorescent and is easily visualizable. Here is this regimen that we discovered. CHRR is an E. coli enzyme. And as I mentioned, the conversion of a CNOB with this enzyme CHRR leads to the formation of MCHB, which is highly fluorescent. This is a cell monolayer, and this magenta color shows that they have MCHB in them. If you look at this figure carefully, you would see that some of the cells have these yellow spots. These are the cells that have been transfected with the CHRR gene. You can see that there are only a few of them. And yet the entire monolayer is showing magenta color. What this shows is that MCHB, after having been generated by a few cells, can leak out of them and can get into the neighboring cells. This is called the bystander effect, which is very important for any gene-directed therapy, because no method of transfection of cells by a gene can transfect 100% of the cells. This figure here shows you the spheroid giving you the same results in a more realistic setting. What this shows you here is that you can visualize it directly without having to sacrifice the mouse. And the magenta color, as you can see, is confined to the tumor. This ability to see the activation of the drug in a particular locale provides the way of finding out, without having to kill tons of mice, whether the activation of the prodrug is confined where you want it to be confined. This shows you the quantitative aspect of it. MCHB is much more fluorescent than is doxorubicin, and it is fluorescent down to 2 nanogram of MCHB per gram tumor. So it's really highly sensitive. So now this slide deals with the way in which MCHB kills the cells. We ran a number of tests, DNA binding, cell cycle assays, determining of apoptosis, caspase, and such. The results are that MCHB binds DNA and arrests cells in S phase. It exposes phosphatidylserine on the membrane surface of the cells, which are treated with it, induces caspase 3, and these are indications of apoptosis. It co-localizes with and depolarizes mitochondria. So the mode of MCHB action is mitochondrial-mediated apoptosis, which kills both growing and non-growing cells. As I said, the importance of non-growing cells I will be addressing shortly. This is the original configuration of our testing of the Prodrug Regimen that I have just described. We were not, at this stage, capable of testing it in a real-world situation. All we wanted to know was whether this treatment would be effective in mice. So we used an artificial situation. What we did was to implant 41 xenografts that endogenously expressed the enzyme HCHRR6, which converts CNOB into MCHB. The rest of the curves in this graph are controls and we can ignore them. The point is that when the enzyme is present in the tumors and CNOB is being administered intravenously, you can see that the mice survived the longest period of time. It took about 60 days for all of them, or nearly all of them, to die. The mice receiving the full treatment survive much longer than those that are controls, and it has very favorable PK/PD Cmax and AUC 0 to 24 is all very favorable. And also the Phoenix Nolan algorithm predicted that low milligram per kilogram of mice dose is sufficient. So these are all quite favorable results. And we have improved upon it, as I have been saying already, by improving CHRR, improving gene delivery, using directed gene delivery vehicles for systemic administration. Okay, so now enzyme improvement. As I've been saying, this is quite important. The enzyme that we improved is CHRR, which is an E. coli enzyme. So let's first see what this enzyme is. We discovered this when we were studying with the Department of Energy ways of bioremediating carcinogenic metals like chromium-6, which is highly carcinogenic. Later, we found that it can remediate many other such metals like uranium-4. And these metals are abundant in the Department of Energy-based sites as well as in Superfund sites. So DOE is very interested in finding ways of detoxifying them. We found that CHRR is widespread in bacteria, and its physiological role in the bacteria is to defend them against oxidative stress. And it can activate CNOB and tretazicol and many other prodrugs. This was something that we found later. And so we call it an enzyme for all seasons. We discovered by X-ray crystallography that it is a tetramer with a flavin mononucleotide pocket as its active site. So now, to improve it, obviously we used the standard procedures, Garrett-Brown PCR, and then a new method, an algorithm that was developed at the Stanford Business School. Here is the screening part of it. Because MCHB is fluorescent, it became really quite easy to see cells containing mutated genes. And of course, the ones that generated the more intense magenta color were the ones we were interested in. Then we did crystallography, which told us the reason in some of these procedures like DNA shuffling or PCR, why the improvement occurred that we saw. The gist of it is that if the dimer interaction is strengthened, the enzyme is improved. Number two, if the FMN active site is enlarged, then too, it is improved. You can see some of the ways in which this can be done. These asterisks show you the changes that we made, which made the enzyme 100-fold more active. And then also we humanized it by codon substitutions. Then the gene delivery vehicle. We didn't want to use viruses. Nanoparticles are great. They have their own problems, but indeed, nanoparticles, I think, have a great future. Already, there's quite a bit of progress made with lipid nanoparticles. But these extracellular vesicles, also called exosomes, they are nature's own delivery system. All the cells in human, animal bodies make them, and there are ways to transfer information even at a distance within the body. Therefore, they are likely to be non-immunogenic. Tetraspenin CD9 mediated fusion results in direct injection of the cargo in recipient cell cytosol, so the effect is rapid. They are small, therefore avoid the reticular endothelial system. They can extravasate through the vessel fenestrations, which is helpful for treating cancer. They avoid liver, lung entrapment and the resulting toxicity, and they avoid the lysosomal endosomal pathway. In fact, it has been shown that HEC293, human embryo kidney 293 exosomes, are harmless in mice. I think the EVs derived from patients' own dendritic cells, for example, would certainly be completely non-immunogenic, and many stem cell EVs would be non-immunogenic. Here we are looking at EVs in TEM. The size is 30 to 100 nanometers, and they contain the three proteins that ascertain that these indeed are EVs. This was done by differential centrifugation. Gene delivery has generally focused on DNA, but we question whether mRNA might be better. There are two issues here: safety. DNA poses the risk of insertional mutagenesis, which mRNA does not. The second one is the speed and efficiency and durability of expression. In this respect, DNA has the advantage that it can generate several copies of mRNA, whereas one mRNA is one mRNA. But DNA, in order to do that, has to enter the nucleus for transcription, while mRNA is directly translated upon cytosol entry. DNA enters the nucleus during mitosis, and its entry and gene expression are therefore delayed, especially in non-growing quiescent cells, which do not have mitosis. The question is, how important are quiescent cells in cancer? This question has been addressed by a number of people. Rapid growth of cancerous cells make many of them nutrient deprived. They grow away from the blood vessels of the tumor, so therefore become nutrient deprived and enter quiescent cell phase. But this phase is reversible, and therefore curing of cancer requires killing also non-growing cells. That was the reason I've been emphasizing non-growing cells earlier. There is direct evidence of the importance of non-growing cells in cancer. These people, Shibumoto and Strafford, used cytochalasin B, which blocks cytoplasmic but not nuclear division. When they scored the multinucleate cells, total cells and all that, they concluded that up to 67% of tumor cells can be quiescent. So it would seem that based on this, mRNA would be superior to DNA for cancer, for treating and hopefully curing cancer. Confirmation of this is provided by Okamura et al.'s work. What they did was to treat malignant melanoma by injecting Bex gene, which encodes Bex protein, which promotes apoptosis. If you look at the diagram on the left, this is when DNA was used in nanoparticles to treat this disease. And this is when mRNA was used. And you can see that mRNA is superior. They also did tunnel analysis. It is clear that the apoptotic index is higher with mRNA gene delivery. This really is convincing evidence that mRNA is more suitable as gene delivery vehicle when you are dealing with cancer. Now mRNA is fine, but now how to put it into EVs? EVs have been easily loaded with small RNAs, siRNAs and such. But putting mRNA in them had been a problem. We tried and others tried. It just did not work. Nevertheless, in collaboration with a commercial company, we succeeded in constructing a special plasmid which used the zip code. And what is zip code? This is the common sequence found in mRNAs that are native to exosomes. Exosomes or EVs contain cellular content, which depends on from what cell they have originated, many of which include mRNAs, and these mRNAs contain a common sequence. When we used that, we were able to construct this plasmid. When this plasmid was used to transfect cells, they generated EVs containing the HCHRR6 mRNA, which is what we wanted. But plasmids have problems, and if you want to take this to the clinic, which we indeed hope to, there is some concern as to what the administration or injection of plasmids into the patient might do. So now we use in vitro transcribed HCHRR6 mRNA, which is used to directly introduce this mRNA into the cells, which eventually will make the EVs. In fact, this method proved more effective than the plasmid-based EVs, as I will presently describe. So now mRNA is into the EVs. The question now is how to make them directed. Remember, directedness is an important part of our investigation because we want that upon systemic administration, the EVs can find their way to the cancer that we are trying to cure and inject that mRNA specifically into them so that the activation of the prodrug is confined to the tumor. In order to construct a protein that can target the HER2 positive tumor, we made a hybrid protein containing lactaserine C1C2, which can bind to the phosphatidylserine that is expressed on the surface of the EVs. This was combined with ML39 SCFV antibody, which has a very high affinity for the HER2 receptor. And then this leader sequence, its purpose is to enable the protein to migrate to the surface of the exosomes and a histag for purification. This is the predicted structure of this protein. The blue is the C1C2 domain, yellow is the SCFV, and then there is the linker, the red one. So the loading of in vitro synthesized mRNA into the EVs, these are the steps towards clinical transfer. As I mentioned, we didn't want to use plasmids. So this is one step. The other is using Trazicab or CB1954 instead of the CNOB, although the CNOB has been very useful in initial studies to make sure that we are using methods to localize activation of the prodrug in the tumor alone. But Trazicab has the advantage that it has been subjected to clinical trials, and so it's safe. Human dose is established. But putting IVT mRNA into EV requires several steps. And mRNA being unstable at every step, we had to make sure that it was capable of activating CB1954. To determine if CB1954 or Trazicab is being converted to its toxic product, whose name appears here, the really big one, which is abbreviated as MPNQ. That has been difficult. It requires very complicated, labor-intensive HPLC and such. So then we hypothesized that since HCHRR6 can activate both CNOB as well as CB1954 and many others, but that's not really important here. Point is that it can activate both CNOB and CB1954. So therefore, if at any of these steps, we could show that the translated product of the mRNA could generate easily visualizable fluorescence upon CNOB addition, we could assume that it would also have the capability of activating CB1954, and that proved to be true. This we called MCHB test. These are technical details of how the IVT mRNA was translated. Translation in vitro used standard kits, and we added this cap and poly-A tail to ensure the stability of mRNA and facilitating its translation. These IVT are transfected cells, again HEC293 cells. They were transfected using polyethylenilamine. They generated EVs that exhibited this EVHP, the protein that I have just showed you. We extracted the RNA from these EVs, ensured that it was functional, quantified it, CT values and all that. What we found is, with all the proper calculations, that there were 3.3 times 10 to the minus 2 copy per EV by this IVT method, compared to 2 times 10 to the minus 4 mRNA copy per EV by the Plasmid method. So the IVT is far more effective than the Plasmid one, and the reason I think ought to be obvious. So instead of 5,000 EVs to deliver one mRNA copy with the Plasmid-made EVs, we needed less than 50 with the IVT-made EVs. This shows that the mRNA is in the exosome. RNA treatment doesn't make any difference. Here are a number of tests. ELISA showed that EVs need to display EVHP to bind HER2 plus cells, showing that EVHP is functional. Flow cytometry showed that it binds exclusively to HER2 positive cells. The experiments with actinomycin D and cyclohexamide showed that upon introduction into the recipient cells, the expression of the gene is insensitive to actinomycin D, showing that no transcription is needed. Had it been DNA, this would be inhibited. So it is indeed mRNA, and cyclohexamide, which is a protein synthesis inhibitor, eliminates it. What that showed is that these loaded and directed EVs do indeed transfect mRNA, which then is translated into the recipient cells and then is able to confer on the recipient cells the capability to activate the prodrugs. Since activating prodrugs is referred to as GDEPT, we decided to name these loaded and directed EVs EXO-DEPTs. These are the main points of the data. It's just for your convenience. You can go over it. And if you have any question, we can discuss them. Another thing we found is that the IVT EXO-DEPTs had higher and sustained expression. The blue is P-EXO-DEPTs, the plasmid, and the red is IVT EXO-DEPTs. You can see that it is more durable and higher expression. How efficient is the transfer from EVs to the recipient cells? We use flow cytometry at different times. There are a lot of calculations, and I don't want to go into the nitty-gritty of it. The point is that at 6 hours, each recipient cell received 5 copies, which corresponded to 50% transfer efficiency. And at the higher incubation time is 80%. That's really impressive, and it can be attributed to the fact that this scFv has high affinity for HER2. Here is the regimen with which the EXO-DEPTs and the prodrug CB1954 were administered to the mice for the preclinical test that we are now discussing. The black arrows show the administration of the EXO-DEPTs, the yellow ones that of CB1954. As you can see, the EXO-DEPTs were administered only twice, on day 0, just at the start of the experiment, and on day 4. The prodrug CB1954 was administered 6 times from day 1, day 2, etc. And the last one was at day 7. The point to note here is that the entire treatment ended at day 7. So this slide shows you the outcome. Mice were implanted orthotopically with HER2 positive xenografts, and then different regimens were tested. These are all controls, and we need not focus too much on them right now. As you can see, the most effective treatment is when the EXO-DEPTs are administered with CB1954. All of this is systemic. We did not inject directly into the tumor. Once again, this is HER2 positive breast cancer implants in mice. You can see that with this full treatment, there is just slight growth and then no further growth at all, which may suggest that the tumor was really killed because the last administration, the last treatment was on day 7, and yet for the next 34 days, there was no further growth of the tumor. All right, fine. The tumor is very likely killed. The EVs go elsewhere too. They temporarily colonize pancreas, spleen, and such. The scFv antibody can also recognize mouse ERBB2 expressing cells, which are not unusual. They do have quite a few of them. And so it was possible that the EXO-DEPTs introduced mRNA also elsewhere. And when you injected the prodrug, The damage was generalized, damaging other organs and tissues. And so we did a very thorough examination, hematology, serum chemistry parameters, which are listed in here, organ histopathological evaluation, organs listed here, hematology. Okay. In all of these tests, we found absolutely no side effects. Any side effects that you saw, they've been shown to be due to CV1954 alone, and they're only transient, so not a serious issue. Serum chemistry, again, no side effects. Now, let me refer you to another recent study done using DNA for gene delivery. They used a rather inventive technique of making a hybrid enzyme that could activate two prodrugs, genciclovir and tretazicon. And they used mini circles, miniaturized plasmid containing this gene. But these were non-directed exosomes. They found 54% tumor kill. Now, when we compared it with the number of genes that they had to inject compared to what we had to inject, it is clear that 2.75 times 10 to the 2 fewer copies of the gene were needed in our case, despite the fact that the injection was systemic, whereas their injection was directly into the tube. This highlights the fact that using mRNA for gene delivery is much more effective than using DNA. Remember, in this study, they used DNA for gene delivery. All of this work was done in immune-compromised mice, but we think that in immune-competent situation, which we are exploring now, which can be even more effective because HER2 tumor ablation evokes a strong anti-HER2 immune response. And this therapy does contain human elements, the EVHB protein as human. So there can be an immune response against these things in mice. But the lack of general toxicity that I just showed you very strongly suggests, I think shows, that the EXO-DEPTs are arrested by the HER2 tumor. Therefore, any immune rejection against these EXO-DEPTs would actually be directed against the tumor, which is where the EXO-DEPTs are confined. Therefore, it might even bolster the killing of the tumor. This is being tested now in immune-competent FBNJ mice, which spontaneously develop HER2 breast cancer, driven by an oncogenic form of human HER2. EVs can cross the blood-brain barrier. We have some results, but very preliminary, that they can affect the spread of the cancer to the brain. The technology is generic. As I mentioned, we have concentrated on HER2-positive breast cancer, but many other diseases overexpress receptors and ligands. And so you can find SCFVs, other targeting moieties to make EVs directed against prostate cancer or many others, transferrin for Alzheimer's disease, for example. Now, one issue with exosomes is that making them is not easy, and there are quite a few efforts underway to increase their yield and make it easier. There is this one in which nitrogen cavitation was used and sudden release of the pressure increased the yield 16-fold. They found that if you incubated the cells making EVs with liposomes, the exosome generation was enhanced, and certain sulfhydryl blocking agents and reducing agents very significantly increase the generation of EVs. So these are preliminary beginning studies, and I think sooner or later a method will be found to do this. Towards clinical trial, these are all standard procedures. We are thinking of phase 1 clinical trial, and some of the details are given in here. These are all standard, so I won't go into it, all dictated by FDA. Now, one final word about NMP, lipid nanoparticles, for mRNA delivery. Lipid nanoparticles have the advantage that they themselves stabilize mRNA. I told you mRNA is highly unstable, so anything that can contribute towards their stabilization is important. LNPs are quite easy to make in large quantities, as is now evident with all these anti-COVID vaccines. All you need is lipid in ethanol, mRNA in water, in a microfluidic device, which is available from many commercial sources. As a result, advances have been made to treat cystic fibrosis by using CFTR mRNA with administration by inhalation. And another study, really quite a remarkable study, targeted CRISPR-Cas, and the Cas was encoded by mRNA for organ-specific editing of PTEN. But lipid nanoparticles have problems in your blood circulation. Lipid can activate immune responses in your liver and lungs, but countermeasures are being undertaken. And given the success already achieved, I think a lot of people believe that LNPs hold great promise in mRNA-mediated gene delivery. Thank you.